Welcome back to our channel. Today we're diving into the fascinating world of DNA replication, a process that's essential for life as we know it. So let's get started. DNA replication is the biological process that produces two identical replicas of DNA from one original DNA molecule. DNA and its role. DNA plays a fundamental role in cells. It serves as the ultimate director, encoding the instructions for traits. Given its importance when a cell divides, it must replicate its DNA to ensure the new daughter cell receives an exact copy. This brings us to our topic. DNA replication, the process of making more DNA. Where and when does DNA replication occur? Where? In eukaryotic cells, DNA replication takes place in the nucleus. However, not all cells have a nucleus. Prokaryotic cells, for instance, lack one. Despite this, both types of cells undergo DNA replication, though the process varies slightly between them. When DNA replication happens before cell division, ensuring that each new daughter cell inherits a complete set of DNA. In eukaryotic cells, this process occurs during interphase before mitosis or meiosis. Key enzymes in DNA replication. Several enzymes are essential for DNA replication. Enzymes typically have names ending in ase, indicating their role in catalyzing biochemical reactions. 1. Helicase, the unzipping enzyme, unwinds and separates the two DNA strands by breaking hydrogen bonds. 2. DNA Polymerase, the builder, synthesizes a new DNA strand by adding complementary nucleosides. 3. Primase, the initializer, creates an RNA primer to guide DNA polymerases to the starting point. For Ligase, the gluer joins DNA fragments, ensuring a continuous strand. The steps of DNA replication. Replication begins at a specific sequence, known as the origin of replication. 1. Helicase unwinds the DNA, separating the two strands to single-stranded. Binding SSB proteins prevent the strands from reattaching. 3. Topoisomerase alleviates supercoiling, ensuring smooth strand separation. For primase, adds RNA primers, marking the starting points for replication. 5. DNA Polymerase builds new DNA strands, following the base pairing rules. Adenine pairs with thymine T, guanine G pairs with cytosine C, understanding DNA strands and directionality. DNA strands are antiparallel, meaning they run in opposite directions. One strand runs 5 feet to 3, while the other runs 3 feet to 5. DNA polymerases can only synthesize in the 5 feet to 3 feet direction. This creates two types of newly synthesized strands. Leading strand continuously synthesized. LEGGING strands synthesized in short segments called Okazaki fragments, which are later joined by ligase. Final steps in proofreading after replication. Each new DNA molecule consists of one original strand and one newly synthesized strand, making the process semi-conservative. DNA polymerase proofreads to correct errors, ensuring high fidelity in replication. Why is DNA replication important? Understanding DNA replication has led to life-saving medical treatments, such as therapies that target cancer cells or pathogenic bacteria by disrupting their DNA replication process. For those interested in exploring this topic further, we encourage you to check out additional resources. Stay curious.